Hello, this is Joyeeta Dutta from the University of Toledo, Department of Bioinformatics, Proteomics, and Genomics. I'm going to present my poster entitled Identification of Common Gene Expression Patterns Associated with Systemic Sclerosis. On the screen, you can see the poster. In this study, I have been guided by my supervisor, Dr. Sadiq Khudair. He is also a co-author in this study. We studied systemic sclerosis, which is an autoimmune disease, and it is a complex one. I would like to tell a little bit about the biology of the disease. It, like other autoimmune diseases, it is also very complex. Its pathological features include skin and visceral fibrosis, vascular alteration, and autoantibodies against various cellular antigens. It involves thickening and hardening of the skin tissue and other connective tissues. Presently, there is no cure for this disease, but luckily, a variety of treatments can ease the symptoms and improve the quality of life of the patients. So that's why we are interested in this disease. We are looking for some biomarker which can be used for treatment and maybe for early detection of the disease. So we used gene expression data for our study. We used 5 data sets, 3 of which were from skin tissues and 2 of which were from blood tissues. The sample source of the data were taken from systemic sclerosis patients with affected skin and blood, and that of the healthy controls. And we downloaded from the data from the gene expression omnibus, which is also known as GEO database. What we did is we made a list of the top 1000 differentially expressed genes for each of those data, and common genes among those lists were identified using the R program. As a result, we got a list of 31 genes which were present in all the 3 lists of the skin, whereas we got 40 genes which were shared among systemic sclerosis blood samples. Interestingly, we didn't find any gene which were common between these 2 lists. I mean, the common genes in skin and the common genes in blood. When we compared these 2, there was no common gene between these 2 kind of tissues. So this is kind of an interesting finding, because we expected there might be some genes which would be common in these 2 studies, but we didn't get any. So among those 31 genes which were significantly differentially expressed, the most differentially expressed genes were ARPC1B, BASP1, C2, COL1, 5A1, COMP, and GMFG. The functions of this list you can see on the screen. I have made a chart of the functions for these genes and the most DG genes from blood also. But for skin, these genes are mainly involved in promoting homologous recombination repair in response to DNA damage and regulation of immune response, and possibly they are associated with autoimmunity. This finding is similar to the findings of the study by Zhu et al. They found some different genes, but their functional similarity is evidenced with our finding. For the genes found in blood tissues, AHR, APL4C, B3GLCT, and BACH1 were significantly the most differentially expressed genes, and they are part of a protein metabolic pathway. Their detailed function is also listed in the chart. You can have a look over there. We have further plan for studying the lungs datasets for systemic sclerosis. Following the similar method, we are planning to compare the lungs differentially expressed genes in the lungs with these two findings and see whether there is any common gene between lungs and blood and between lungs and tissue. So we hope our study will be able to find some biomarker for systemic sclerosis and may be used as a means of early detection and also treatment for the disease. Thank you all for listening. If you have any question, please don't hesitate to ask me. Thank you.